So they gave me the luxury of talking about whatever I wanted to talk about. And so I picked something very dear to my heart, and that's imaging the cellular universe. And so what I want to do tonight is to first give you a clue as to how imaging technologies developed. And I want to talk about the birth of cell biology, because microscopes really made it possible for cell biology to happen. And so I want to talk about the microscopes and people that made it possible. Now, I thought that was a great idea when I first decided to write this lecture. And I soon realized that I had collected enough material for a whole semester course. And so I decided you didn't want to sit here that long, so I had to cull it down and try to pick out the most important details. And so what I've decided to do is just focus on those technologies that have been extremely useful to cell biology. Let me just fix this. So the tools that have enabled us to see cells in unique ways. Louder? I'm not sure I have a control on that. Do I have a control on that? This has been tricky.
Turn our volume on.
Is that any better? Okay, great. And so I want to talk about how those microscopes have revealed cell structure and function. And in fact, everything we know about cell structure has been revealed by microscopes of one sort or another, and much of what we know about cell function has been revealed by microscopes. So they allow us to see things we can't otherwise see. And one of my favorite things is that new microscopes generate new findings, which generate new questions, which generate the need for new imaging tools, which generate new findings. And you can see there's a feedback. There's a constant driving. So the two fields, microscopy and cell biology, are closely intertwined and wedded together and developing simultaneously. And then I want to end by talking about where the future will take us. So seeing things you can't otherwise see. You look at this and you look at a thumb holding a pen. Now some of you with very good eyes still can see that there's actually a hair on top of that pen. And if you have an imaging aid, such as a magnifying glass, you can see that there's something else on there. There's this pink dust mite. And if you look closer and closer, you see that there's this little yellow thing. And it turns out that when you look closer, it's ragweed pollen. And then there's other things on there. There's a purple lymphocyte and a red blood cell. And then another little light thing, this little pink thing, which is yeast. And we can look, there's even more. There's a little green bacterium, E. coli, and yellow bacteria, Staphylococcus. And there's even more. There's an Ebola virus in the purple there. But we're not done. There's also rhinovirus. So we're imaging things you can't otherwise see by using microscopes. And with this particular time sequence here, this animation, we've gone from what you can see with your eye all the way up to what you would see with the electron microscope. And so this is what I want to do tonight, is show you how we image these things. Now, it's not just magnification that counts, it's resolution. And resolution is defined as the distance between, the shortest distance between 2 points that can still be distinguished as separate entities. So here you can see these are clearly distinct red circles. But here it's hard to know if it's one circle or two circles. And so this would have a lower resolution or worse resolution than with this sort of instrument. So that's what we're talking about with resolution. It's not just magnification. You can make something bigger and bigger and bigger, but if it's not resolving two spots as distinct spots, it's not better resolution. And so the slides to date show what you can see with the eye, and you can see all the way up to this hair. You can see the hair with the naked eye. But to get to the cells and the bacteria, the cells are about 1/10 the diameter of a hair cell. You're going to need microscopes. And the players to date have been the light microscope and electron microscope, as shown by this little slide from Zeiss, who hasn't learned that there are more microscopes than this. And not only do you need good microscopes, but you need people who are capable of imaging cells with a different eye. And I want to quote Dan Mazia, a cell biologist from whom I took a course in 1983 at Stanford's research station in Monterey, an excellent cell biologist who studied mitosis and learned a lot. And as he said, the gift of the great microscopist is the ability to think with the eyes and see with the brain. And he was one who did that, and also with a great deal of enthusiasm. He was quite a mentor and a teacher who could show enthusiasm. I think he was in his 70s when I took the course from him, and he was as enthused looking through a microscope as a freshman. So this is the cellular universe. And this is a model from textbooks, and this is what we know about cells today, and it's been learned from microscopes. So at the center of the universe, you have the nucleus, and there are a bunch of planets in this universe. There are endoplasmic reticulum, things called Golgi, things called centrioles, and things called mitochondria. So these are all the different planets in the universe. So how do we know that they're there, and what do we know about their function? Well, let's start with the earliest players. We've all heard about Antony van Leeuwenhoek, who's referred to as the father of microscopy. And we know that in about 1673, he saw red blood cells, he described protozoa and bacteria, and lots of little things he called animalcules. He did this with a very simple microscope, a single lens. This little microscope was about 2 inches, and it's just much like a magnifying glass, but this was new to them. Didn't have the best resolution, but it was very easy to use, just like holding a magnifying glass. And these are the sorts of things he saw. And can you imagine the excitement he felt when he looked into a glass of water, for example, or a dish of water and saw things moving around? Well, it might have been exciting, but it might also have been frightening to see little things moving around in the water he was drinking. Now, the downside of being a microscopist back in Leeuwenhoek's day was that you had to draw what you saw. There wasn't film. I would have been out of a job. I can't draw. These are Leeuwenhoek's drawings, little bacteria and bacteria here. And he also looked at blood cells, and you can see these little circles that he called blood cells. So although he's referred to as the father of microscopy, there were people working before him. There was Marcello Malpighi, who was a physician and professor of medicine, and actually the founder of microscopic anatomy and first histologist. So since I'm in a department of anatomy at UCSF and I teach histology, I like this guy, and he's important to me, so I have to point out that he actually studied things like skin and kidney and liver and various embryologies before Leeuwenhoek was made famous. And he used a little more complex microscope. This is what's called a compound microscope, and that's only because it has more than one lens. But with that microscope, and this isn't much taller, it's just a little bitty microscope about this high, but he was able to see a number of things that hadn't been seen before. And then there was Robert Hooke, who actually coined the term cell. He was the first one to use this term, and it was a while before it stuck, but it did. And he did that because he was looking at a thin piece of cork, and in the cork he saw all of these little box-like structures. And they reminded him of the small rooms in a monastery, which are known as cells. So he called them cells, and that's the beginning of cell biology. He also used a compound microscope and wrote this wonderful book called Micrographia in 1665. Now what you notice with this microscope is something that we haven't mentioned yet, was they didn't have bright light bulbs, and they were using candles and oil lamps and very inefficient light sources. But they did a very good job with what they had, and some of them even used sun that they would reflect off the specimen and observe the shadow to identify what they were seeing. So those days were a little bit more difficult. And then there's Henry Baker. He wrote 2 wonderful books, The Microscope Made Easy and Employment for the Microscope, an interesting way to phrase it. But he, in this book, described many ways of preparing, examining, and preserving specimens, and described his surprising discoveries. And he was actually the first one who did sort of experimental science with a microscope. He would look at different solutions and describe what he saw in those solutions. And these are some of the chapters in his book. Of the animalcules in fluids, of eels in paste, of rainwater and other waters, wonderful topics. And what he did was he studied things like pepper water. He'd take a dish, put pepper in it, and compare the growth that he saw or the movement of things in that water compared to no pepper in it. He saw that after a couple of days, they'd get this skin on top, clearly containing the bacteria, and he would look at that and see massive amounts of animalcules. He didn't know what they were. And he studied things like hay water, straw, grass, put all of these different things in the water. This is the first cell culture media. You know, he was beginning to develop methods for culturing cells. Eels and paste, they were just, I'm sure, long, skinny bacteria, but he called them long, slender, wriggling animalcules that grow daily until you can see them with the naked eye. Now here he also described water that drains from dunghills is thronged with animalcules. You and I wouldn't find that very surprising today, but that was an important finding, to find lots of animalcules in the dunghills. So it's a wonderful book. And these are his drawings, or drawings from his book. Some were Leeuwenhoek's also, showing all of the different animalcules. And he's a good artist, so he got away with this. I'd be out of a job. Now, the next couple centuries were centuries of physics rather than biology, and I can summarize those since I'm focusing on biology tonight by saying lots of improvements in lenses. The next important thing for studying cells was developing the ability to take pictures so that you could record the image that you saw in the micrograph. Now, that wasn't easy to begin with. Imagine trying to focus to take the picture. Here's your microscope, and here's the operator down here. And you've got all of this distance. When you look at a microscope, you typically want to focus it. This is not easy. So there's a string here. I don't know if you can see it, but he's got a string attached to the microscope with which he can focus the microscope and take the image. So it wasn't easy, but it was definitely an improvement over drawing. And if you were to take a replica of Leeuwenhoek's scope, which Brian Ford did, and look at bacteria, they would look like this, with this exact same microscope, but recorded on film rather than drawings. And you can see that Leeuwenhoek did a good job. They look like bacteria. And the red blood cells. These are red blood cells seen with this replica and seen in his drawings. Much easier, though, to take the picture. And the next major improvement was methods for fixing, sectioning, and staining. When you use these simple microscopes or any light microscope today, things that are too thick are impossible to see through. So you have to find ways of making them thinner so you can see through them and record an image. And that requires fixing them, sectioning, and staining so that you can see them. And so with a variety of different dyes and testing various things, he developed methods for staining, and these are blood cells. Now that they're stained, you can clearly see the difference between the blood cell types. The neutrophil has multiple lobes, the monocyte looks like a kidney bean, and the lymphocytes have a nice clear round nucleus. These techniques are used today, developed in the mid-1800s, and they're still used today. Sectioning tissue, here's a piece of liver with cords of cells radiating from this central vein. Looks very different from the section of a kidney where you have these glomeruli, these circular things where the kidney filtration occurs. Looks different from the intestine where you have these large globular cells that secrete mucus to make it easier for the food to go through. And the lung, an airway here with lots of fluffy cells around the periphery. These techniques are still used today. If you get a biopsy, the physician will compare the normal versus the abnormal using these very same staining techniques. Not much progress in that area. The next major development was phase contrast. And those of you who were in Chris's talk this morning learned about phase contrast, so I'm not going to go into the physics of it. But what it did was enable the examination of live specimens and unstained tissues. Live cells are transparent. You can't see them, so you have to use some tricks, and he used phase contrast. Now, what I love is that he developed this in 1930, and he went to Zeiss in 1932, the microscope company, and tried to convince them it was a good thing. But one of the older, wiser scientists said, if this really had any practical value, then we would have invented it a long time ago. Sound familiar? He got the Nobel Prize in 1953. And I have to tell you, this is the most frequently used microscope technique in cell biology. You can't see cells without this. You have to use this to watch your cells growing in culture. So it's had a very major impact on studying cells. And this is what it allows you to do. If you look at something in a a microscope without the phase contrast, this is the cell. This is what the cell looks like. If you look at it with phase contrast, it pops out. And so you can see this that you couldn't otherwise see. Now with the modern microscopes and modern cameras, you get a very nice image of cell structure. Lots of clearly discernible nuclei, the center of the universe, and lots of organelles and particles out here around the nuclei. And this one's dividing. The cells are getting thick when they divide, so it's harder to see through, and you can see clearly dividing cells. So a very major development. But what were those granules that we saw in there, and what else is in the nucleus? Are there ways that we can see those particular structures? So the next approach was to use better resolution techniques. And the way to get better resolution is to use a shorter wavelength. So they went to the electron and developed the electron microscope. And this is the electron microscope developed by Knoll and Ruska, physicists who developed this in 1931, and Ruska got the Nobel Prize in 1986. The first biologist to have a major impact on the study of cells with the electron microscope was Keith Porter. Took the first electron micrograph of a whole cell in 1944, discovered many of the cell structures, ER, cilia, microtubules, lots and lots of those planets inside of the cell universe. He also realized that when you look at a whole cell in electron microscopes, the electrons don't have enough energy to get through the whole cell, so you had to develop methods for staining them. You need a couple hundred thin sections to get through the whole cell, so he developed methods for sectioning them, developed the first ultramicrotome, the first fine sectioner, and was also the founding father of the American Society for Cell Biology and the Journal of Cell Biology. So he had a major impact on our understanding of cell structure. And this is the first cell he saw. It's a whole cell, and you can see cytoplasm has a lot of interesting structure in it. This nucleus here is too thick to see through. And you can see that it's also a montage, a compilation of a number of small pictures to get it all into the field of view. But this was the first cell, and he saw these long wormy structures out in here, which he called the endoplasmic reticulum. But how do you get higher resolution information and see through such thick structures? And that's with sectioning. So when you section, you get a lot more information. And you see that the universe is surrounded by this double-layered membrane. That's the outside protecting part. And it's composed of lipids, which are kind of waxy and keep water out, and lots of proteins and glycoproteins. And these make channels and transport mechanisms that allow only what you want inside to get inside. Other things that you could see with the electron microscope, structures, planets of the universe that were identified, the endoplasmic reticulum, edge of the nucleus here. And when you color code an electron micrograph, you can see the nucleus. The endoplasmic reticulum is where proteins are made. These little red dots are the ribosomes that make that protein. And you can see it looks just like the model. And the Golgi apparatus. This is where proteins go after they've been made to have sugars put on them. The sugars target their destination in the cell. And when you color code that, you get a structure that looks like this, that looks a lot like the model. And mitochondria. The mitochondria are the energy source of the cell. Your ATP is made in here. That's where you get energy to do all cell functions as well as your whole physiology functions. And when you color code them, they look like this. And you can see they look just like the model. So that model really did evolve from all of the studies of electron microscopy. But to see things on the cell surface that are filamentous, like the extracellular matrix, you have to use another EM technique where you make a replica of the surface after etching away water, and then you put that replica in the microscope. So you can get even higher resolution. And that means that you're seeing very, very small fibers in here, about 3 nanometers in resolution. So it's much, much, much smaller than the diameter of the human hair. The next major thing was Hollywood. We could now do video microscopy. We could study live cells. You have to use the phase contrast imaging, but you could study them live. And you could learn things about cell function and cell differentiation. How does a cell become a heart cell or a neuron? And so this is a movie that was taken long ago, looks like early Hollywood, and you're watching this neutrophil. The neutrophils are the cells in your body that fight infections, bacterial infections, etc. And it's homing in on this yeast cell, and it's going to engulf it. So we learned something about the function of neutrophils. It's also a bit like human behavior. The big guy eats the little guy. And you can watch him engulf it. And here's another similarity to human behavior. If you put a lot of yeast cells on the plate or a lot of food on the plate, the neutrophil will engulf all of the yeast cells. And what you can see here, where this one yeast cell is located, is the granules inside of the neutrophil that are degrading it. They're filled with lysozyme, enzymes that break down the yeast cell and digest it. So it will eat and digest all of these yeast cells. Now this was something that wasn't possible to observe until we had the ability to image these cells alive. We can also watch the neutrophil eat bacteria. So here's the neutrophil here. These are red blood cells. So you've taken a little bit of droplet of blood, put it on a slide, and you add some bacteria to it. And here's the bacterium. Neutrophils are the guys that I call the garbage cans. They run around and eat up all of the bacteria. And you can see it's going to chase the bacteria. It's persistent. Finally gets it. But it's not full, so it's going after the next one. So we learn about neutrophil behavior. We can also learn about different cell types. And this is a muscle cell, a single muscle cell. It's not a whole heart. It's a muscle cell that's been put on a substrate that has contractile properties. And so you can watch that muscle cell contract in a dish. Now, to be a heart, it has to, all of the muscle cells have to beat in synchrony. But here you can study the contractile properties of this cell. The next major development was what we call Nomarski interference contrast or differential interference contrast. And this detects optical gradients and converts them into intensity differences. And it's also a great way of imaging live cells. And what you now see compared to this phase contrast is more of a topographical view of the cell. Surface structures stand out very nicely. And this is what epithelial cells would look like. The nuclei, the cell surface very clearly, and organelles inside. But the best invention was when Robert Day Allen and Nina Allen were trying to prepare a lecture to show to their class. And they'd been studying cell motility, something that wasn't really believed to be possible. And they wanted to be able to show the motility they were seeing in the microscope to the whole class. So Nina got the idea of hooking up a camera, a video camera, to the microscope. And it turned out they could see more than they did through the microscope. So it's called video-enhanced microscopy. And she could see lots of organelles moving along the microtubules. Now the microtubules are these linear structures that are a little bit hard to see here. But you can see them, and you can't see them just through the regular microscope at that time. And here's another picture where you can clearly see the microtubules. They're very small, 25 nanometers in diameter, and you can see this freeway system. There's cars moving in both directions. And it looks like some of them are playing bumper cars. So that was a really exciting finding. You could actually demonstrate cell motility. And then we found that there are actually different motors. There are motors that take it to either the plus end or minus end of the microtubule, which is a polymer with actual polarity. And you could see an organelle moving in this direction. And what this demonstrates in here, one going towards the plus end, this one going towards the minus end. And this one's going to change its mind and go back. So organelles have both plus-in directed motors and minus-in directed motors. And so rather than this structure that you saw in the model based on electron microscopy, rather than these being just architectural proteins, they're actual freeway systems that send proteins and organelles around the cell. And those microtubules are found in neurons, the long extensions from neurons, which is what these are in culture. So the neurons in your brain, the cell body, this large structure, will make proteins that perhaps your toe needs. And so it's going to send them along these long processes, along microtubules, out to the periphery. So you can study these cells, which look very different from a heart cell, in the dish and watch the long extensions growing. And then with DIC, you get an even better topographical view. And you can see all the little fine extensions on these neurons. Wound healing. This is an image of the fin of a very small fish. And there's a wound right here. This is a blood vessel, a vein. And what you're going to see is the cells of your immune system, which are flowing through your veins, want to respond to that wound. And you can see them crawling out away from the vein towards the wound. And when you zoom out a little bit more, you can see that these cells, which are fibroblasts, aren't moving, but the white blood cells are moving out to the wound to respond and deal with the damage. You can also look at the cell cycle. If you put cells in culture, they like to attach. At first, they're going to wiggle around a lot until they get attached to their substrate. And they'll calm down a little bit once they start attaching. There they go. Cells don't like to be isolated. Their blood cells don't mind it, but the other cells like to be attached. And you can see now that it's attached, and these, they like to have contact. They're a little bit happier when they touch each other. And then you can see it round up and divide. So you can actually watch cell division.
What is the ratio time contrast?
The,
In other words, how long does this take?
it takes about 24 hours for a cell to divide in culture. Yeah, so it's highly sped up.
This is sped up.
Yeah, very much sped up, yeah. And so when the cell is dividing, it's actually going through the process of separating chromosomes. So the first thing that happens before the cell is going to divide is it duplicates its chromosomes, then it separates the chromosomes and produces two daughter cells. So you can see there's the duplication of chromosome material, they line up, and then they separate. Well, you can watch that with DIC imaging. And these are the chromosomes. And you soon will see, in some cases, the obvious doublets. I'm not sure if the screen, yes, you can see some of the doublets. These are actually duplicated chromosomes. And they're going to line up on what's called the metaphase plate. And they're doing that by those microtubules, those things that had organelles running up and down them, are also involved in cell division. And the microtubules run out and grab a chromosome, grab onto it. And until all of those chromosomes are attached to microtubules, they're just going to keep dancing around like this. Once they're attached on this plate, the microtubules will then pull the chromosomes apart. Still getting all the chromosomes, and then it will pull them apart and then take them to the opposite poles. So you can learn about cell division using this approach. Chemotaxis. How did those cells know to go to the wound site, for example? Well, we now know that there are chemicals that attract them. And these little dictyostelium are being attracted by a chemical in that pipette. And by this very simple approach of putting a chemical in the pipette and seeing whether or not a cell responds, you can learn what attracts various cells. And you can perhaps aid certain processes. The neutrophil chemotaxis. Remember, we saw the bacteria being eaten by the neutrophil. Well, when neutrophils are in your bloodstream, they're rounded, They would have to be rounded to flow through the bloodstream. But they also respond to chemicals. So when you have a wound or when you have a foreign object, a bacterium or a yeast cell, they secrete chemicals. And that chemical attracts the bacteria, attracts the neutrophil to the bacteria or to the wound site. And so now you've released the chemical here, and the bacteria starts crawling towards that chemical in the pipette. Now you can actually tease the bacteria. And they will respond. So you can use these approaches to determine what chemicals cells are responsive to. The next thing is the development of fluorescence microscopy. And this allowed us to look at specific proteins by exciting that protein with a fluorophore and emitting at a specific light. And what it allowed us to do is stain things, for example, the nucleus here. We stained it with a dye that will fluoresce when excited. And that allows you to label a specific protein, and then you can superimpose that image with this phase contrast image and see where the nuclei are. So you could do that with a number of different proteins and find out where they are in the cell. But what really made the news with fluorescence microscopy was the invention of the scanning laser confocal microscope. And that allowed you to get very sharp planes of focus. Cells are thick. You look in the regular microscope and everything glows, and it's a very fuzzy image. But when you use the confocal, distinct sections of the cell will stand out very nicely. Now, this is one of those tools that had a long lag time. Minsky first demonstrated it was possible in '57, but he didn't have a way to record the images. It was very complex. And then in '87, finally, these folks, Amos and White, made the first confocal that could store images. What made this possible was development of lasers, computers, and microelectronics. And then in the '90s, it had an enormous impact on the cell biology community. Thousands and thousands and thousands of papers have been published using the confocal microscope. And that's because you could label specific proteins. Here we've got the actin labeled in green, microtubules in red, nucleus in blue. So not only could you look at a whole cell, but you could look at where a specific protein is in that cell. Very powerful technique. The next thing that hit the market was GFP, and this wonderful little jellyfish is responsible for that. It's a jellyfish that was found up at Friday Harbor, and in fact, I took a course up there in 1983 before we had the GFP protein, and we would go out in the boats at night, and you could put your paddle through the water, and you get this wonderful view of sparkling little fluorescence in the water. All the little jellyfish would emit a fluorescent color. So it was great fun going out and looking at what looked like fireworks in the water just by running your paddle through. Well, it turned out to be a very powerful tool. Osamu Shimomura isolated it way back in 1960 and determined which region was responsible for the fluorescence. But it wasn't until 1994 that Preischer and Chalfie demonstrated its ability to tag proteins. So what you can now do is insert the gene sequence for this little glowing protein, which he's showing here, a bottle of glowing protein, put it into the protein of interest, and you can track it around live cells. So it allowed you to look at specific proteins in the living cell. And this is one of those proteins tagged with GFP. And this is a whole collection of cells. And you can see the GFP is around the surface of the cells. They're dividing. You can see them getting smaller and smaller. And eventually, the protein goes into the nucleus. So you could track the movement of proteins. At one time, it's at the surface. The next time, it goes into the nucleus. And so this revealed a lot of new information about protein function. And what we learned was that proteins don't stay in one place. The static view showed them either always on the surface or in the nucleus or somewhere. They move around. And when they move, their function changes. This is a picture of a field of cells with microtubules labeled in green. And this is a protein that travels along microtubules out to the periphery of the cell. This centrosome is called the microtubule organizing region, and that's because it organizes all those microtubules and sends this protein out to their tips. and there's the actual microtubules in the cell. So you get a lot of information about a specific protein in a live cell. This shows the actual motor that's moving the protein along the microtubules. This is the kinesin light chain, and it moves proteins towards the plus ends of those tubules. And so you can actually see the linear paths by the direction of the protein movement. So thousands of papers have now been published tracking proteins in live cells using this GFP protein. Other things you can study. This is a listeria-infected cell. Listeria is a bacteria that causes a very severe infection. And you can see the periphery of the cell, and these little black worms are the bacteria, worm-like structures, are the bacteria running around the cell. And they divide very quickly, and as they divide, obviously you get more and more bacteria, and it's a very deadly disease. And you can see some of, one of these will divide. These guys are trying to push out to an adjacent cell, but it's more comfortable inside, so they go back in. This one's dividing. Now, how are they moving around? They're moving around by using what's called an actin comet tail. They take over the actin in the cell, the normal cell actin, they polymerize it into a tail, which you can see in red here, and that pushes them around the dish. So it was thought for a long time that bacteria were really clever. They could learn how to create a mechanism in the cell to kill the cell. Well, we showed that naturally, cells naturally have the same mechanism. These are vesicles in the cell that are coated with a protein called PKC, and it's causing the initiation of actin comet tails to push the organelles around the cell. So it's not that the bacteria were so clever they created a mechanism. They were even more clever. They just hijacked the mechanism. The mechanism was there. They just hijacked it. And it's a very efficient way to move around a cell, rapidly divide, and decimate the cells. But all stuff that's learned by studying live cells. So one thing I like to say is that no single microscope can answer all questions. We need a combination of microscopes, and let me show you an example of that. There's Leeuwenhoek's drawings, the same red blood cell seen with the replica, seen with differential interference contrast. You can now see some of the fibers that are part of the clotting factors. And seen with an scanning electron microscope, where you get higher resolution views of the structure of a red blood cell. These cells don't have a nucleus, so that's why they have this sort of concave center. They're strictly going to carry oxygen around and deliver oxygen to the tissues, so they don't need a nucleus. They've finished making things. And when you see it in the transmission electron microscope, this is actually in a capillary. So you've got an endothelial cell with microvilli lining the surface, and the concave view of the red blood cell here and a surface view here. But then when you take something called a laser tweezer, you can learn more about the red blood cell, and you can see that it's actually pliable. So laser tweezers allow you to push around the cell attached to the microscope with DIC. You can learn that blood cells are very pliable. And that's because they have to sneak through the very small capillaries and get to every portion of the body. So different techniques tell you different things. All of them look remarkably similar and very convincingly look like red blood cells. You can also look at those neutrophils, the neutrophil that was chasing the bacteria. Seen in sections, seen in a thin section in the electron microscope, might look like 4 nuclei, but it's actually one highly folded like you see here. Seen with a scanning electron microscope, crawling neutrophil with lots of bacteria on its surface. And another view with a scanning electron microscope showing its correlation with the red blood cells. So a combination of microscopes can reveal a bigger picture and a bigger story than can one single microscope. Well, if no single microscope can answer all questions, but I've just shown you a powerful tool chest of microscopes, is there any need for any more microscopes? Do we have enough microscopes? Well, when you use a light microscope, you have limited resolution, but you can look at live things. When you look at the electron microscope, you can get very thin structures at very high resolution, but they're dehydrated, they're fixed, they're embedded in plastic, they're sectioned very thin. To look at a whole cell, you have to collect a couple hundred thin sections and then try to reconstruct them. What would happen if you had one microscope that could look at a whole cell at better resolution than the light microscope? That would be nice. What would you do if you got better resolution? You could see the whole thing in 3 dimensions. You could also localize proteins like you can with both the light and electron microscope. Well, that would probably be a good thing. So now we can talk about imaging with X-rays. And this is something that we like to say can bridge this gap between the very fine, thin stuff in the electron microscope and the live, thick stuff in the optical microscope at poorer resolution, and fill in the gaps, look at whole cells, fully hydrated, and get 3-dimensional and what we call isotropic resolution. Well, that was made possible by development of synchrotrons, like we have here at Berkeley. They have much brighter light, which is important. Leeuwenhoek used candles. That clearly wouldn't do for X-ray tomography. So we have the synchrotron to give us very, very bright light. There were a number of players here, and the synchrotron X-ray microscopes, the first one in 1970 by Günter Schmal right here in Germany, and it was a transmission X-ray microscope, and Janos Kurtz here developed the scanning X-ray microscope in New York at Brookhaven National Lab in the 80s. And then we built the ALS, and right here in Berkeley, Dave Atwood developed the first transmission X-ray microscope in Berkeley. And that's with the Center for X-ray Optics, and he's at LBL and UC Berkeley. And we owe them a great deal of gratitude for allowing us to use their microscope to show you the next sets of imaging tool biology. So this is the picture of the X-ray microscope. It looks a little different than your standard little tiny 2-inch microscope. It's probably about 20 feet long here. And this was built by Werner Meyer-Ilse, Hector Medecki, and Dave Atwood. Just diagrammatically showing you that it's much longer, but the specimen is going to sit right in here, which is right about here on this microscope. What made this possible, in addition to the synchrotrons, was nanotechnology. The ability to focus the light depended upon the development of these lenses. You can't focus X-rays with glass or with magnets like you use for the electron microscope. So they went to a structure that's very similar to the Fresnel lenses that you see in a lighthouse. You can diffract the light and get a very, very bright beam to focus on your specimen. They're very tiny, 63 microns in diameter for an objective zone plate, and very precise, getting very symmetrical rings and finer as you go out. And so this, the nanotechnology, the development of synchrotrons, really made major advances in the ability to use X-rays to image cells. And so this is what you see. Looks much like an electron microscope. You see a nucleus, you see lots of structures in the cytoplasm, but there's some major differences. This is a whole cell, it's fully hydrated, no fixatives, no stains, no contrast enhancement reagents. Remember the first cell that Keith Porter saw? The nucleus was so dense you couldn't see through it. We can see through it with the X-ray microscope. And we can see lots of organelles in here. So that was extremely exciting to us. But then it became clear that that's very thick material. So how are we going to see everything we need to see in that thick material? Everything stacked up, you have to resolve it. And the way you do that is by doing CAT scans, much like a CAT scan of the knee or head or any other structure that you've gone to a hospital to have. Rather than doing a CAT scan like they do in the hospital, we're going to do the CAT scan of the single cell. Very same principle. So you take multiple images from multiple angles and reconstruct them. What we did was start with yeast cells, that little yeast cell like the bacteria was eating, sorry, like the neutrophil was eating. And this is a cell that's dividing, and you can see the division plane right here. So the mother cell and the daughter cell out here, and that's looking at the surface view. If you want to remove the surface and look at internal structures, you can do that, and you can see lots of organelles inside of the yeast. And because we're using the X-rays, we can make this quantitative, and you can see all the different structures and color code them based on their absorbance. So it's now a quantitative tool that you can use to look at the cell structure. This one is not dividing yet. You can see the nucleus in blue here and a large vacuole and lots of little droplets. Lots of information in here. So now you can get better resolution than with the light microscope, not quite as good as with the electron microscope, but it only takes 3 minutes to do this. And to get one single section from the electron microscope can take a week. So that's a big advantage. And you can also look at things such as cell division. These are a different type of yeast that are dividing. We're just going to focus on the division plane here. And you can see the differences between the cell division in these different yeast. Now, this is not published, so you're actually looking at something that the rest of the world hasn't seen. This is the cell division. You can see early cell division, the contractile ring is just forming. Hard to tell from the projection image like you would see in the light microscope, but when you do the tomography, do the reconstruction, you can see this one's early in division, this one's a little bit later, and this one's more complete. So we can learn new things. Fully hydrated cells takes 3 to 5 minutes to collect the whole tomographic data set. And then you can compare what's happening during cell division, and we can also localize proteins. So we can look at protein, the distribution of proteins just like you can with a light microscope, but at better resolution. And that's an extremely powerful thing to do. These are bacteria, the very small bacteria like the neutrophil was eating, 2 microns long, ½ micron in diameter, very, very tiny. And you can see a whole cluster of bacteria in this tube. And here are just 2 of those bacteria. So extremely small structures, 2 microns. If the human hair is 25 microns, 50 microns, it's significantly smaller. So let's compare those with other techniques. Leeuwenhoek's drawings, the bacteria seen with the Leeuwenhoek replica, bacteria seen with the light microscope, seen with X-ray tomography, sectioning through this particular tomographic reconstruction, showing you all of the detail inside of the bacteria. So lots of internal structures. That same sort of bacteria seen in the scanning electron microscope, which looks at scattered electrons and gives you a surface view. And then a bacteria seen in the electron microscope. So this is one single section, where here with the tomographic reconstruction, you get many sections. And because this is fully hydrated and no embedding material, the structures stand out more than with a plastic section because the plastic interferes with seeing the structures. So we now have another powerful tool in the toolbox of microscopes. So where can we go from here? What does the future bring? Well, we have this wonderful model of cell structure, a lot of which came from electron microscopy, and we're getting more and more information about proteins from various biochemistry and molecular biology tools. Can we superimpose that information? Can we take the information from all these protein interaction networks showing where all of these different types of proteins are, superimpose them on the cell tomogram, and get a 3-dimensional view of the protein distribution in the cell? That's our goal. That's what we want to do. And we're going to start doing that with this recently established National Center for X-ray Tomography, where we've been generously funded by the National Center for Research Resources and Department of Energy, Biological and Environmental Sciences to build a new microscope dedicated to biological imaging and to make it available to the biological community so we can start using this tool to develop our proteome map. So here's our collection of microscopes over time. You'll notice that there was a lot in here as far as biology is concerned, a big gap, and then a huge boom in imaging techniques. And I think it's still taking off a lot of new imaging techniques. A number that I didn't have time to discuss, as I said, it would take a whole semester course. But I tried to pick those that have had the most impact, and one that I think will have a huge impact in the future, the X-ray tomography. And to close with another quote from Dan Mazia, not only is the gift of the great microscopist the ability to think with the eyes and see with the brain, but deep revelations into the nature of living things continue to travel on beams of light. And I think we will continue to see great things from microscopes. So I should thank all of the people involved. This is our new National Center for X-ray Tomography. Mark Legros is the physicist who's building the new microscope and largely responsible for all the data that you saw, with help from a nice, wonderful group of people who are supporting him and me in developing this new center. The Center for X-ray Optics, who allowed us to use their microscope to collect all of the data that I showed you from the X-ray microscope. Advanced Light Source, Engineering Division for all their help with building the new microscope, University of California, San Francisco for employing me, and all of the important funding agencies without which this couldn't have happened. And I should also give some credit to the wonderful web pages that had a lot of information on them that I was able to borrow, and textbooks that provide a lot of those video clips that we use as teaching tools. And I also want to thank you for your attention.
Thank you, Carolyn. Questions? Yes.
Got microphone here?
So technically, what are the key factors for you to keep those cells alive under X-ray exposure, especially if you want to focus the X-ray?
We can't look at live cells with the X-ray microscope. They were initially live but rapidly frozen. So we're looking at frozen cells, and you can do fixed moments in time, but they're frozen.
Could you go back one screen and let us copy some of those?
Sure, absolutely.
While you're copying those down, any other questions?
Do you detect, for X-ray tomography, do you detect the X-rays by their scattering out of the direct beam, or rather than by absorption?
We're using absorption because we're imaging in a region of the X-ray spectrum where the carbon and nitrogen found in our proteins absorb much more than surrounding water. So it gives us this natural contrast.
Using the K edge or something?
Yes, that's right, between the K edges of carbon and oxygen.
Anyone else?
Corey?
Thank you.
Could you say another word or two about these things that swallow up the yeast cells?
Those are your neutrophils. Yeah. They're white blood cells. It's the critical blood cell that is, you have immune system blood cells, the white cells that are responsible for fighting infection and for killing all foreign invaders. So if you poke yourself with a rusty nail, you're going to get a lot of bacteria in that site, and the neutrophils will come charging there to eat up all the foreign invaders. They're loaded with enzymes that are specifically killer enzymes, and they will kill anything that they get near. That's their job.
More questions?
I have a question concerning the images how you produce in the end, because the stage from a microscope in the end to this 3D animation is probably required for quite a bit of computer vision or computer graphics.
I'm sorry, say that again?
The stage from the image you get from a microscope to the end, in the end you want to have a 3D image which is animated, and you want to see isosurfaces, and you want to see through them. This requires quite a bit of computer graphics or computer vision. So what kinds of algorithms do you use, or how difficult is it for you, and how much do you work together with computer vision folks?
We stole all of the algorithms from the electron microscopy community. We started developing electron tomography before we did. And so we were able to use all their algorithms for the actual alignment. It's not trivial. You have to put little markers in there and then align it and then do back projection reconstructions. We do it all on either PC, I think PC or Mac now. I think we can also use the Mac. And then we use a commercially available program called Amira for all of the isosurface and all of the volume rendering and stuff like that. We are not specializing in developing computer programs for processing or displaying. We steal and buy commercially available. Not steal, the electron microscopy community has made it available free of charge, so you can just go to the web page and download it. So it's very nice.
Of course, we all know of the explosion in genetic information, genetic engineering, and all that sort of thing. Do you see any synergy between this type of microscopy and that information? I mean, is there any way the data can feed into each other?
Exactly where we want to go with it. You know, the product of the gene is the protein. And so we can localize the proteins with that technology. I didn't have time to show how we do it. Rather than a fluorescent tag, one that glows a nice pretty color, we use a metal tag that you can see in the X-ray microscope. So we can localize all the gene products. Eventually, it would be nice if we could also locate different genes. And there are ways of doing that that we're developing too. So we're developing all of the methods to do exactly that, locate genes, count the number. Because this technology is completely quantitative, you can actually count the number of proteins, labeled proteins, count the number of genes, and those sorts of things, once we develop the technology to do that. It's just a matter of developing labels and figuring out how to get those labels on the structures. I didn't have time to show a slide, but there was a quote from Peter Satir, who was one of Porter's students, and in his biography, he said that when they developed the electron microscope, one of the major challenges was learning how to prepare specimens and how to interpret what they were seeing. And we're going to have that same challenge of preparing methods and interpretation techniques. Yes?
Carolyn, you showed a lot of dynamic images, but then, of course, what you're doing now with tomography is in fact a static image. Do you envisage perhaps an additional dimension that goes into this dynamic aspect of some very minor systems?
What we plan to do is have a light microscope right by the X-ray microscope so that we could track a GFP tag protein, for example, in the live cell, and then take that very same cell to the X-ray microscope and get the better resolution imaging. So we can't do live cells in the X-ray microscope, but it'd be complementary, and it would actually enhance what you learn from the light microscope, because one of the things that's happening is we're seeing thousands and thousands of papers published in Cell Biology where they talk about the distribution of proteins. And there are only so many things you can say from a light micrograph. They're on the cell surface, they're in the cytoplasm, they're in the nucleus, and maybe ER, maybe mitochondria. The resolution when you do those approaches is probably 300 nanometer resolution. A protein's only 4 nanometers. So when they say a protein's co-localized, it might only be 300 nanometers away from a protein rather than really co-localized and interacting. So we can fine-tune that, get down to better resolution information about how close proteins are to each other. Are they interacting? That's the next really challenging field of cell biology, is protein networks and protein interactions. Biochemistry is coming up with hits for 1,000 interactions between 2 proteins. You can use one technique and get this 1,000, use another technique, get another 1,000. Maybe there's 30 that both techniques identify as interactions. So what's really happening in the cell? So we can use that combination of the GFP tracking the live protein and looking at its distribution at better resolution with the X-ray microscope. Second best to doing live cells.
other questions? Am I missing anything? All right,
Thank you.
on behalf of the UC, I wanted to thank Carolyn again
Oh,
for
my.
the amazing talk.
Thank you.
Nice bottle of wine.
Thank you. Lovely.